This might be the next bio company to take off. Stay tuned. You really don't want to miss this one. How's it going, guys? In today's video, we want to share with you a company that's been on the radar and we've been picking up shares of within the last couple of months. We've been buying slowly just because it's a newer company, but I think with the research that we've been doing, I think this company can really be like a picks and shovel type play for the biogenomics industry. This company's been out and a lot of people have made videos on it. I've been doing more and more research on the company and I'm really starting to like it. The company I am speaking about is BNGO Genomics, ticker symbol BNGO. There's several factors why I like this company. One of them is the price. It's really cheap compared to some of its competitors, if you want to call it that. But it also does something different than its competitors, and we're going to go through that information today. Just to let you know, guys, this is not financial advice, but I do share my opinions of what we're doing here, and it's worked out for us, so maybe it can work out for you. We do have a current position at an average of 1019 on BNGO Genomics. In the beginning, we had a bigger position, but this used to be a penny stock not too long ago. And we would keep buying and selling, and at one point I stopped and I thought about it and I built a small position and I said, wait a minute, this stock is could be a game changer. Okay, it can be a game changer, and I'll tell you why shortly. But we do hold about right here 815 shares of this stock, together with in other portfolios as well. We do hold close to 1,200 shares. Okay, 1,200 shares of this stock. I am planning to keep this for a long time hold. I am not going to be selling this one here. It has too much upside for me to think about selling it. So from this point on, I will be holding this stock. If you look at the chart here, I'm going to go through the 3-month chart. Not too long ago, late last year, it was only at 55 cents. Okay, but it has came up, and the high point here was about $13-14, I think it was. And since then, it's settled out a little bit. If you want to look at the week chart, it did take a dip last week, and I was buying last week, and then it came back up this week. Okay, and right now, currently, it's on an uptrend. So, it's a good time to buy. If I could suggest for you to buy this stock, it's actually at a good price right now. But if I could suggest to you, buy it anywhere between 10 and 11.50 even. A good price, and I think for the future upgrowth, I think you have a tremendous opportunity, and I'll tell you why. So taking a look at their website here, BNGO Genomics, Saphyr reveals genetic drivers of severe COVID-19 suspectability. Okay. They recently had a big study not too long ago, and they found out that they were pretty much asking themselves, why is it that some people that get COVID, get really sick, and then some people that don't, some people that get it, they don't get sick at all. They don't even actually feel any symptoms. What this product does, and if you see here the Saphyr, is that it reads the DNA. And I'm going to play a short video for you just to give you a better understanding, because I'm not a doctor and I don't know how to explain it too well. But what I do know is that it reads the DNA and it gives you information in regards to your DNA and why it reads it that way. Okay, so I am going to be playing a video for you, and here you go.
This is Saphyr from BNGO Genomics. With it, you can directly image the genome in high resolution to detect variations such as deletions, duplications, translocations, and inversions, known as structural variations, with sensitivity as high as 99%. The process is simple and automated to enable routine use in disease research. Structural variation commonly occurs in a broad range of diseases, including neurodegenerative diseases, intellectual disabilities, and various types of cancer. In fact, the first targeted cancer therapies were based on gene fusions, a type of structural variant. Given the role structural variants play in both understanding and developing potential targeted therapies, it is important that structural variants are accurately identified or called. To do this, there are two fundamentally different yet complementary approaches: sequencing and whole genome imaging. In short-read sequencing or next-generation sequencing, DNA is read in short segments of about 150 base pairs, and the read sequences are compared to a reference genome. It can characterize small variants, but structural variants are often tens of thousands of base pairs or longer, so short-read sequencing is not an accurate way of calling them. What's more, 2/3 of the genome is repetitive, and structural variants in repeat regions are often invisible by whole genome sequencing. Whole genome imaging with the Saphyr system takes an entirely different approach to understanding the structure of DNA through imaging. It has proven to accurately identify structural variants ranging from 500 base pairs to whole chromosome lengths with sensitivity as high as 99%. First, native ultra-high molecular weight DNA, multi-megabase pairs in length, is isolated. Fluorescent labels are enzymatically attached to these ultra-long molecules of DNA at a specific sequence motif that occurs throughout the genome. The resulting label patterns are unique for each section of the genome. The full length of the DNA remains intact during isolation and labeling, preserving the architecture for the next step, imaging the genome. The labeled DNA is pipetted into the Saphyr chip and placed in the Saphyr instrument. In the chip, the DNA is uncoiled or linearized across thousands of parallel nanochannels and imaged, revealing its fluorescent label patterns. By capturing these images in repeated cycles across the hundreds of thousands of nanochannels on the chip, all the images necessary to assemble a map of an entire genome are captured. Algorithms extract molecules from the images, and then bio-nano algorithms construct consensus genome maps. Bio-nano Access software also allows easy visualization and filtering of SV calls using our built-in control SV database. From the thousands of SVs found in any human sample, you can remove variants present in controls and narrow down your SVs of interest to a handful with just a few clicks. This is all enabled due to the extremely long molecules isolated and imaged with the Saphyr system. Want to use whole genome imaging for your next discovery? Go to bionanogenomics.com to learn more about how you can call more structural variants with higher accuracy and sensitivity.
So guys, with this system, the Saphyr system, doctors, people, scientists can read DNA and its structure and kind of, in simple terms, be able to take out certain things in your genes. So say, for example, if you're sick and you have, say, for example, you have cancer, they will be able to read your genes, and along the DNA strands, they will be able to read that and pick that up off the reading and tell you exactly where that reading is at. And therefore, a doctor or whoever's in charge of taking the next step can go ahead and replace that gene or that strand and be able to basically cure you. Now, there are several other companies that are in the bio sector. And remember, guys, we had spoken about this sector being one of the biggest sectors in 2021. That was one of the biggest ones, okay? And that's why I'm so intrigued with this company. And the reason why I say it's a picks and shovel of this sector is for this reason. Bio-nano systems provides essential genomic information no other technology can provide to help advance greater understanding of genome biology. Bio-nano's next generation mapping combines proprietary nanochannel arrays with optical mapping to image extremely long, high molecular weight DNA in its most native state. This technology enables unparalleled structural variation sensitivity, genome assembly, contiguity hundreds of times that a short read sequence alone, and the accuracy to correct sequence-based errors. So guys, with this system, you're pretty much able to read the strands and therefore detect what problems lie within your body. I think with BioNano, I think we're going to be able to use this as the picks and shovels, because it's basically going to be able to read, okay? This system here, the Saphyr, this will be able to read the strand. And since there's so many bio companies coming out, some of them including CRISPR, some of them including Pacific Bio, they're just coming out left and right. I think a lot of these companies will need this company to be able to read the DNA. And another thing that I also noticed is that in its pricing, Saphyr system starting at $150,000, and then it's $550 per genome, $450 per genome with 240 genome bundle installations and training included. So I believe they also charge per reading. So if you look at it that way, guys, and you look at this sector, I really do believe with all the diseases that are out there right now, science itself is trying to find a way to better alleviate all these problems. And in my particular case, I have been affected with cancer. Not myself, but I do have family members that have gone through it. I have family members that have passed away from it. And I think BNGO can actually revolutionize this whole sector, and it'll be an extremely important aspect of it. Guys, I think this can really explode to a lot more than 10 bucks, okay? I see it going anywhere between $20 to $30 in the short term, and can really go as high as $100 long term. I think if everything works out and everything that they say is real and correct, I think they have a bright future, guys. With that, guys, I'm gonna let you guys go. I don't want to take too much time, but I do want to give you this information. And if you guys found any of this information informative, please like and subscribe, and also follow us on Twitter. I keep repeating it because I want you guys to follow us and get the latest information at Investing OG. We've been on there, we've been giving out information, we've been giving out plays, and they've really worked out. With that, guys, have yourself a great day, and I'll see you on the next one.